All right, let's get started. My name is Richard Kondo, and I'm Director of Business Development at AnaBios. Thank you for joining us today for another round in our webinar series on investigations of biomedical issues and how they relate to human disease. As a quick intro, AnaBios is a unique contract research organization and a biotech company based in San Diego. We recover human organs from ethically consented donors and use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We've essentially redefined first in human. And working with AnaBios is as close as you can get to a clinical trial without actually doing a clinical trial. For those working in heart and want to learn more about our cardiac platform, a heart brochure is available for download in the handout section of the GoToWebinar panel. Today's presentation is the third talk this year devoted to the heart. We'll be taking a summer break from the webinar series and returning to it in the fall. So please stay tuned. I also want to thank Gary Watkins, Director of Marketing at AnaBios, for putting together this webinar series. If we could move to the next slide. So I want to announce that we're extending the application deadline to July 15th for our Translational Award, which is an award for academic researchers worth $15,000 for the obtaining of human tissue samples from AnaBios. As an organization, AnaBios is committed to enabling human-focused drug discovery in areas high unmet medical need. This award is designed to assist the academic researcher in their translational project by providing access to high-quality human tissue samples. In addition to the winner, the 10 runner-up applications will get a 25% discount for the future purchase of tissue samples. So please, if you're interested, submit an application for this award. So next slide, please. So with no further ado, it's my great pleasure to introduce today's speaker. Dr. Najah Abi-Gerges is VP of R&D at AnaBios. He holds a PhD in cardiac physiology from Paris University in France. Prior to joining AnaBios, he was involved in drug discovery programs at AstraZeneca. He has over 20 years of experience in the pharmaceutical industry. He's an innovative leader and has made substantial contributions to drug approvals and research across multiple areas of cardiac physiology and pharmacology, resulting in over 50 published articles and in novel paradigms to advance cardiovascular translational scientists. He's also editor for the Journal of Pharmacological and Toxicological Methods. Today's talk is entitled Arrhythmogenic and Antiarrhythmic Actions of Late Sustained Sodium Current in the Adult Human Heart. Thank you.
Thank you for the introduction. Welcome everybody to today's webinar, which is about arrhythmogenic and antiarrhythmic actions of late sustained sodium current in the adult human heart. Late sodium current has been investigated in rabbit heart, guinea pig heart, porcine heart, and dog heart. These studies have shown late sodium current to contribute to action potential, to sodium homeostasis, to arrhythmogenicity, and to dysrhythmia. Additionally, late sodium current has recently been nominated as one of the cardiac currents of the Comprehensive In Vitro Proarrhythmia Assay Initiative. An increase in late sodium function has been proposed to play a critical role in long QT syndrome types 2 and 3, in atrial fibrillation, in heart failure, and in hypoxia oxidative stress. Therefore, a reduction of late sodium current could have therapeutic benefits. Such therapeutic benefits have been shown in 2 recent clinical cardiac safety studies. Data from these studies demonstrate that inhibition of late sodium current with ranolazine, mexiletine, or lidocaine may offer protection against abnormal ventricular repolarization. The therapeutic benefit of inhibition of late sodium current have also been shown in the management of atrial fibrillation. For example, as shown on the left-hand side of this slide, synergistic reduction in atrial fibrillation burden was seen in patients treated with moderate dose of ranolazine plus a reduced dose of dronedarone. Dronedarone, an antiarrhythmic drug that blocks sodium, potassium, and calcium channels, and it also inhibits beta-adrenergic receptors. The right-hand side presents a second clinical study. This study shows that when data were combined from patient groups treated with 2 doses of ranolazine, a reduction in recurrence of atrial fibrillation was observed. Taken together, this data suggests a possible role for inhibition of late sodium current in the treatment of atrial fibrillation. As you can notice from these introductory slides, the specific contribution for late sodium current in the genesis or suppression of arrhythmias in human cardiomyocyte and tissues is still unknown. To address this knowledge gap, we conducted the study we are presenting today to define the role of late sodium current in human action potential morphology, in human calcium dynamics, in human contractility, in human arrhythmogenesis, antiarrhythmic treatment, and protection against drug-induced proarrhythmia. Next slide provides information on the method we used to conduct the study. We used isolated atrial and ventricular cardiomyocyte from normal donor hearts. We also used atrial and ventricular trabecula from normal donors and donors affected by atrial fibrillation. We used voltage clamp to record late sodium current and current clamp to record action potential of cardiomyocyte. We used fluorescence technique to measure calcium transient and bright field imaging technique to measure contractility of cardiomyocyte. We used organ-based technique to measure force of contraction of atrial and ventricular trabecula. To modulate the function of late sodium current, we used ATX2 to potentiate the current and mimic long QT syndrome type 3. We used E4031 to block IKR-HERG channel and mimic long QT syndrome type 2. We used ranolazine and GS967 as inhibitor of late sodium currents. We first investigated the properties of late sodium current in ventricular cardiomyocyte. We applied a voltage ramp protocol designed to mimic key features of the cardiac action potential waveform. This protocol elicits an inward current that peaks at 0 millivolt. We found ranolazine to inhibit baseline late sodium current. This indicates that the current, when recorded in human primary cardiomyocyte, can be used to determine if novel drugs inhibit non-enhanced late sodium current. Also, we found that ATX enhances significantly the late sodium current and causes a left shift in the current voltage relationship. This effect of ATX is completely reversed by the addition of ranolazine. In summary, this data provide evidence that isolated ventricular cardiomyocyte express functional late sodium current. To investigate a possible role for late sodium current in cardiac action potential, we measured the effect of the inhibitors GS967 and ranolazine. As it can be seen in this slide, we found that the application of either GS967 or ranolazine shortens the action potential and decreases its duration. And also it reduces beat-to-beat variability in action potential duration at 90% repolarization, quantified here as short-term variability STV. In contrast, we found ATX2 to prolong action potential duration and to increase STV. It can be concluded from this data that late sodium current actively contributes to the ventricular action potential duration and kinetics. It can be concluded also that enhancement of late sodium current could have a proarrhythmic effect in a human adult cardiomyocyte. We next wanted to determine the effect of the inhibition of the late sodium current on ventricular repolarization abnormalities. In first series of experiment, as shown in this slide, we found that application of 0.3 micromole of GS967 decreases 10 nanomole ATX2 induced prolongation of action potential and restore repolarization stability by reducing short-term variability STV. Similar observations were made with ATX2 at 60 nanomole, GS967 at 0.1 micromole, and ranolazine at 10 micromole. We also investigated the extent of interdonor variability of cardiomyocyte responses to ATX2 in 4 different donor hearts. As you can see in this slide, ATX2 treatment was found to result in significant APD90 increase and no statistically significant difference in action potential prolonging effect of ATX2 was noted across cells. This data suggests a low interdonor variability of cardiomyocyte response in the human population. In the next set of experiments, we assess the effect of inhibition of late sodium current on repolarization abnormalities induced by inhibition of the hERG channel with the selective blocker E4031. As shown in this slide, we found E4031 to increase action potential duration and STV. We also found that addition of GS967 largely reverses E4031 induced action potential prolongation and restores action potential stability. We also observed that application of ATX2 and E4031 induce early after depolarization events, another predictor of proarrhythmia. The addition of GS967 was found to inhibit this early after depolarization events. In cardiomyocyte, intracellular calcium dynamics play a critical role in the contraction-excitation coupling, and calcium overload can lead to arrhythmias. To investigate the contribution of late sodium current to physiological and arrhythmic intracellular calcium homeostasis in adult human primary ventricular cardiomyocyte, we assessed the impact of late sodium current potentiation or inhibition while measuring intracellular calcium transient. We first evaluated the stability of calcium dynamics in response to addition of vehicle solution. We found vehicle addition not to affect calcium transient. Our data also demonstrate that the addition of GS967 had no statistically significant effect on the calcium transient. Our data also demonstrate that no statistically significant difference in calcium transient across cells from different donor hearts was observed following the addition of the vehicle solution. We conclude from all this data that the calcium transient model and GS967 can be used to assess the contribution of the late sodium current to calcium handling. We found that potentiation of late sodium current with ATX2 has a profound effect on intracellular calcium handling. We found ATX2 to cause a profound delay in the decay of the calcium transient signal and induce triggered ED-like arrhythmic events. We also found that the GS967 and ranolazine to restore normal calcium dynamics and abolish ATX2-induced arrhythmic events. In conclusion, the potentiation of late sodium current leads to abnormal and irregular calcium dynamic, which could underlie the arrhythmia markers observed in our action potential recording, and inhibition of late sodium current restores physiological calcium dynamics. Given the potent effect of late sodium current modulation on intracellular calcium dynamics, we assumed that changes in late sodium current could impact cellular contractility. We found that ATX2 not only increased cardiomyocyte contractility in a concentration-dependent manner, but also leads to the manifestation of arrhythmic markers like after contraction and contraction failure. We also found that the positive inotropic effect of ATX2 is consistent across cells from 4 different donor hearts. This again suggests a low interdonor variability of cardiomyocyte response in the human population. While enhancement of the late sodium current with ATX2 has a positive inotropic effect, we found that late sodium current inhibition by GS967 had no effect on baseline contractility. This results enabled us to use GS967 to determine the contribution of late sodium current to ATX2-induced abnormal ventricular contractility. As shown in this slide, we found that the addition of GS967 restores normal contractility and significantly reduces the manifestation of arrhythmic markers. We also confirmed the role of late sodium current in ventricular contractility and arrhythmogenesis with ranolazine. Next, we assess the effect of inhibition of late sodium current on abnormal contractility caused by inhibition of the Hurg channel with E4031. We found that E4031 causes a small increase in contractility and leads to the manifestation of after-contraction events. We found that addition of GS967 prevents ATX2-induced increase in contractility and restores the stability of contraction. The results we obtained from a human adult ventricular cardiomyocyte prompted us to test whether the late sodium current plays a similar role in atrial cardiomyocyte. We observed that ATX2 induces positive inotropy and arrhythmia markers in adult human atrial cardiomyocyte, and application of GS967 restores normal contractions. Moreover, we found GS967 to reduce the incidence of after-contraction and contraction failure. As you know, isolated adult human cardiomyocyte provide an extremely convenient preparation for mechanistic, biophysical, and pharmacological studies. However, to rule out the possibility that some of our findings may be attributed to abnormalities in the physiology of isolated cells, we evaluated the arrhythmogenic role of late sodium current in multicellular atrial and ventricular tissue preparations from normal donor hearts. In both cardiac tissue preparations, we found ATX2 to induce positive inotropy and arrhythmia markers, and application of GS967 to reverse these effects. In addition, we found late sodium inhibition in atrial tissue to prevent ATX2-induced development of enhanced automaticity, a marker of atrial ectopic activity. Late sodium current inhibitors have been proposed as potential antiarrhythmic agent for the treatment of atrial fibrillation. Therefore, we assessed the contribution of late sodium current to manifestation of arrhythmic markers in contraction relaxation cycles measured from atrial trabecula obtained from donors with atrial fibrillation. We found that in donors with atrial fibrillation, atrial trabecula exhibit spontaneous arrhythmic markers like after contraction and premature atrial contraction. Please note that premature atrial contraction is a marker of atrial ectopy and also is a clinical predictor of atrial fibrillation. These findings clearly demonstrate the occurrence of atrial arrhythmogenic remodeling. We found that addition of GS967 restore normal atrial contractions, reduce amplitude of contraction, and prevent atrial remodeling-induced events of after contraction and premature atrial contraction. Based on findings with our study, we propose the following mechanistic sequence to explain the contribution of late sodium current to abnormal diastolic calcium. Enhanced late sodium current leads to excessive sodium loading. The increase in intracellular sodium inhibits the forward mode of the sodium-calcium exchanger, and eventually the exchanger via its reverse mode, causing an influx of calcium. Influx of calcium will cause spontaneous calcium release from the sarcoplasmic reticulum. Thus, abnormal diastolic calcium dynamics could act as the trigger for arrhythmias, while increased dispersion of action potential repolarization could provide the substrate. Also, our data clearly demonstrates that late sodium inhibition can protect against drug-induced proarrhythmia and provide antiarrhythmic treatment for atrial fibrillation. To conclude this webinar, late sodium current plays a critical role in human cardiac arrhythmias. Inhibition of late sodium current could provide an effective therapeutic strategy. Our study highlights the utility of the human ex vivo heart models for advancing cardiac and translational sciences. Thank you very much for your attention, and I am happy to answer your questions.
Thank you, Najah. That was an excellent talk. So we're opening up this webinar for a Q&A. You should see a questions section in the GoToMeeting panel, sort of towards the below, and you can type in your questions, and I will read them out. And but first, actually, I will start with a question. So, Najah, you indicated that one of the mechanisms by which the late sodium current could lead to an increase in or generate arrhythmias is that it would lead to an increase in dispersion of repolarization. So I see that in terms of when you add ATX2 and you have an increase in the action potential. But if in the absence of ATX2, would you think that, or is there any evidence that there are spatial gradients in the late sodium current, say across the transmural wall, that also contribute to these great dispersions of repolarization and gradients of action potential duration?
Yeah, certainly, Richard. Beat-to-beat variability in ventricular repolarization has been shown preclinically and clinically, to play a role in arrhythmia and arrhythmogenesis. So, we know that myocytes across the ventricular wall, they have potentially may have potentially different expression of ion channel and different properties of action potential. So it could be a possibility, under pathological condition and non-enhanced late sodium current by a pharmacological agent, this difference to potentially may have different late sodium current contribution, and this may lead to some kind of beat-to-beat variability and may play a role in initiation of arrhythmia. But we still need to go ahead and investigate and assess the characteristic of late sodium current in, for example, epicardial myocyte, mid-myocardial myocyte, and epicardial myocyte. We need first to demonstrate that these cells do have different properties in the human heart. So having different type of myocyte in the ventricular wall is still a matter of debate in the human heart.
Thank you very much. And the second question that comes up is, have you is the molecular identity of the late sodium current been established?
Yeah, I mean, for a long time, the belief was that peak sodium current and late sodium current, may come from their function and activity may be related to one channel, but recently there have been some new studies indicating the presence of different type of sodium channel in the heart, including a neuronal sodium channel. So it's still debatable, if it's coming from one molecular entity or multiple molecular entities. So more work needs to be done, to clarify this point.
Okay. A technical question from a listener: is there a method for accurately estimating the late sodium current magnitude from surface ECGs?
It's a good question. I'm not sure, if there has been any clinical study trying, to assess this possibility, but potentially conducting, clinical ECG studies, or maybe some animal ECG studies, with late sodium inhibitors or enhancer, can help to see if there is any kind of fingerprint that can be derived from ECG recording, and by doing analysis, looking especially at the T wave.
Right. Thank you. A question from another listener is: what is the implications of the GS compound's protective effect on the repolarization abnormalities induced by E4031?
Yeah, basically, GS967 and ranolazine are two known inhibitors of late sodium current, and there is little bit of difference in terms of their selectivity based on data published on both compounds, especially from the CV Therapeutics and Gilead teams. That GS967 is more selective, and when you use it, it can allow you, to selectively modulate and assess the contribution of late sodium current compared to ranolazine, which is a known multi-ion channel blocker. So at 10 micromole, this is why we use 10 micromole, and we used in the study 60 micromole to try to look for ranolazine effect when it's inhibiting selectively late sodium versus at high concentration inhibiting other inward current. When it comes to GS967 and E4031, the ability of E4031 to prolong, the action potential, and especially to prolong, the plateau of the action potential, and allowing late sodium, to become more functional and more impactful. And this is where this is how inhibition of this current can help in protecting against drug-induced proarrhythmia. I hope I answered the question as the listener would like.
Yeah, thank you. And of course, everyone can follow up with any answer with further questions in that chat panel or in the question panel. Another question from a listener is regarding the mechanism around the sodium-calcium exchange. So, do you have in a sense an idea about when or at what concentration of intracellular sodium that the exchanger reverses, and whether or not the late sodium current can deliver enough sodium to even reverse the exchanger?
Yeah, basically, actually, we did not measure, intercellular sodium concentration in the presence of ATX2, but what we did notice that ATX2 is really a very significant, modulator of late sodium current. It caused, a significant increase in action potential, duration in calcium, and contractility. Certainly, this very high level increase of or effect of late sodium of ATX2 is certainly leading to a very fast increase in intracellular sodium, and very quickly, the sodium calcium exchanger is reacting to trying to reestablish, a good balance, between sodium and calcium ions intracellularly. And I believe it's happening very quickly, and the level of sodium is rising very quickly. And And when you use ATX2, you don't really need to wait a long time to see an effect. So the change in the sodium exchanger, calcium exchanger functionality, I believe it's happening very quickly due to a very significant rise in sodium. But again, as I said, we haven't really measured intracellular sodium to try to quantify what I'm trying to explain.
Right. Thank you. Another question. Is there a preclinical species, an animal species that closely resembles a human in the extent that the late sodium current contributes to actual potential duration?
Late sodium current has been investigated in multiple animal models, either at the level of single cells or using Longendorf heart preparation, and also doing some in vivo ECG recording. But what was known about the human late sodium current was really very, very limited. So we wanted to investigate and see where human is maybe close to animal situation, or where they may be different. And I mean, you already can see the size of the current at the baseline level, for example, is much higher in a human cells, compared to animal. And also, there is the question of sensitivity and affinity, and the level of concentration you need to block the current in different species. So based on our experience, you need less concentration. You have higher affinity when it comes to the human situation.
Mm-hmm. Thank you. A second question from a listener is very interesting. What do you think it's worth, and would you be interested in actually looking at the late sodium current in myocytes that were isolated from the pulmonary vein, especially in the context of the potential for triggering AFib?
Yeah, I mean, pulmonary vein and its activity, has been proposed to play also an important role in atrial fibrillation. And as we clearly demonstrate in the webinar today, is that we can recover donor hearts with atrial fibrillation. We can record and demonstrate the role of late sodium current in tissue. So yeah, we haven't attempted to go after late sodium current in pulmonary vein, but the pulmonary vein are one of the tissue we can recover when we have the hearts here at AnaBios. So we have the possibility to work on this and to collaborate with anybody interested in doing this. And I mean, you can see from the webinar today that we haven't yet attempted to isolate single cells from the AFib donor hearts. We demonstrate the characteristic of the current in healthy, and then we use the tissue to demonstrate its therapeutic benefit for patient. So this is the next step, to try to go down, find the right protocols, to isolate the atrial myocyte, ventricular myocyte from these diseased hearts, and also, look at the vein, pulmonary vein, and also isolate cells from pulmonary vein to try to understand, how the current look like, what are its characteristics when it comes to a diseased condition. At the moment, we demonstrated its role at the tissue level.
Right. Thank you very much, Najah. Yeah, I would add to that. We do have the capability of dissecting substructures in the heart, so that one can do fairly detailed, at least in terms of doing molecular discovery, as a start of looking for potential sources for arrhythmias, and for its particular atrial fibrillation. So that's a possibility to do, and we are actually engaged with organizations interested in this approach. Are there any other questions that for Najah? Well, Najah, I want to thank you for a great talk, and I want to thank everybody who has joined us today. I want to note that for those of you who may have been, come in a little bit late, that the webinar has been recorded and is certainly available for review at any stage. You can download and listen to the webinar at the AnaBios website. And I want to again thank you for coming, and please join us for the interesting webinars that will be coming up not only in heart but in areas of neuroscience when we restart in the fall. And I'm going to wish you all have a great and not too hot summer wherever you are. Thank you very much.
Thank you, Richard.